So I'm going to be talking about SZMAP today. SZMAP is our semi-continuum approach to looking at solvent thermodynamics in the protein binding site. And I'll be talking today about the way that we think this provides useful information for guiding structure-based drug design. So because this is a calculation of water, it's useful to remember why we need to worry about water, because it would be much easier if we didn't have to. But the reality is protein ligand binding occurs in water, so we have to account for it if we want to understand binding. And it can be informative because water forms the same interactions with the protein binding site that ligands do. This means that ligands have to break these interactions, whether they be hydrogen bonds or van der Waals interactions, and displace the water in order to come in and bind to the protein. But it also means that if we can understand the way that water prefers to interact with the protein binding site, that can be a guide for us as far as how a ligand might beneficially interact with the protein binding site. And lastly, the role that water plays in binding is often non-obvious and complex. And so if we provide tools that will help to visualize or understand how water is influencing ligand binding, we can add useful information to a modeler's toolbox. So what sort of experimental data do we have about water in a structure-based setting in a protein binding site? And so usually this is coming from X-ray crystallography, and sometimes we see crystallographic waters in a PDB structure. But how much do we really know about, say, water position? Does a crystal structure provide us useful information, experimental information about water position? And I think the answer here is really not definitively. Sometimes we see waters in a PDB file, but if we actually look at the underlying experimental data, the electron density, there's no actual density for that water. So we can't really say that that's a position that's supported by experiment. And secondly, even if we do see electron density, we don't know that that density is due to water. Density could just as likely be due to an ion being at that location, or an excipient, or any of the other stuff that ends up in a crystal structure. But what about the energetics of water in a binding site? Well, here I think we know even less. Right? We know, first of all, that the conditions under which the crystal is made are not the same as the assay conditions in terms of temperature, in terms of salt concentration, or several other factors. But more importantly, we can't actually measure the free energy of a water, an individual water, in the binding site. And so even if we can calculate that energy, we can't compare our calculated energy to any experimental value that's meaningful. And so because of these challenges, we've designed SZMAP and its associated program GAMEPLAN to use the calculation of water as an indicator, as a way to inform us about the protein binding site, to give us insight in where in the binding site can be particularly useful to focus, and also to generate testable hypotheses of what sort of modifications to make to a given ligand. So that's what I'll be talking about today. So if we break our approach in computational chemistry to solvent into three broad categories, we have a variety of ways of dealing with solvent, at least historically. And so the first way of dealing with solvent is to just ignore it completely, to treat electrostatics as if they're occurring in a vacuum. So this is completely wrong. It's like looking at the behavior of an astronaut in space and extrapolating from that about how people behave on the ground, right? We know that there are important effects that water has, the screening of charges, the desolvation penalty of removing good interactions with water, and you miss those completely if you treat electrostatics as if it's happening in a vacuum. So secondly, there are continuum solvent methods. And so these methods treat water kind of similarly to how we experience water as humans. Water is this uniform substance that fills a volume, and it has these average properties that we can observe. And continuum solvent methods are actually very good and very fast at reproducing these average effects of water. And they can be, in fact, quite useful in dealing with cases like ion concentration, where even more complicated methods can have trouble. But continuum methods break down when the behavior of water molecules becomes discrete, when the behavior of individual molecules of water becomes important. So this is the sort of situation we find ourselves in when we look in a protein binding site. And then lastly, there's methods that treat water discretely. So this is, explicit solvent MD would be one of these, where every single water is treated as an individual molecule. And so now we can see the behavior of all these individual waters that can be important in a protein binding site. But you can run into other problems. Sampling becomes an issue in an MD simulation. And in the end, a lot of the computational cost is going towards reproducing the average effects of interactions with water that we can get very cheaply with continuum solvent methods. And so SZMAP is a method that sort of sits between discrete and continuum methods, where we're trying to get the best of both worlds here by treating water as an individual molecule when we need to, while relying on the speed and the accuracy of continuum solvent methods when we think it's appropriate. And so the way that we do this is with this semi-continuum approach, where we're going to have one explicit water molecule, the probe water, in the region of interest at a time. And so we place this, say, into a protein binding site, and we calculate the energy of its interaction with the protein as well as with the rest of the solvent area, which we're going to treat with continuum solvent theory, in our case with Poisson-Boltzmann theory. And so for this water, we calculate its interaction with the protein and with the rest of solvent, and then we look at that energy for many different orientations of the water at that position. And so from this, we can build up an ensemble of orientations and derive thermodynamic properties like its free energy or its orientational entropy at that position. And then we can move that water somewhere else and do it again. And so this is how we're treating one water right at the location we're interested in as an individual molecule, and then the rest of the water is treated with continuum. Now, the fact that we can place this water anywhere and sample arbitrarily is a big advantage of SZMAP over other methods. This means that in an area of interest like a protein binding site, we can define this kind of sampling that we want to do. If we're interested in the entire binding site, we can sample systematically through this binding site by doing the SZMAP sampling on a very fine grid. And this gives us a very thorough sampling of the site, which is independent of the environment, whether it's hydrophilic or hydrophobic. This is something that is sometimes a challenge for MD methods, where you don't know if you've sampled hydrophobic sites or clefts well enough. And so this gives you very reliable, reproducible sampling. We don't have to worry about, if we've run this long enough, if it's converged, because it's reproducible sampling. But we don't have to do this systematic sampling, right? We can decide to sample wherever we want. And this means if we're only interested in a sub-pocket here, we can just sample that sub-pocket, which makes for a much smaller region of interest and a much faster calculation. We could decide, well, I want to evaluate the positions of each of these crystallographic waters to see what sort of environment they're in according to SZMAP. Or we could sample by any other set of arbitrary coordinates, and this is something that will come back to be useful later on in the talk. So I've talked about the focus of SZMAP on the problem of ligand design. And so what we've found to be useful for this problem is to look at a specific location at the difference in energy of a SZMAP calculation of two probes. So one being a regular water, and the other being this hypothetical uncharged water. So this is the same water molecule that makes the same van der Waals interactions. It just has no partial charges, and so it can't make any electrostatic interactions. And so when we compare these energies by subtracting them, we can find two situations. So a situation where the water is more favorable than the uncharged water. So this occurs in cases where the water can make specific polar interactions with the protein binding site. So the water makes those nice polar interactions. The uncharged water can't make them because it doesn't have any charges, and so the water ends up more favorable. And so by default, we're going to color those areas yellow. Alternatively, you could have a situation where the uncharged water is actually more favorable than the regular water. We're going to color those areas purple. And that can occur in cases where there are no good, strong polar interactions for the water to make. And so when the regular water goes from bulk, where it can make nice hydrogen bonds with other water molecules, and finds itself in this place in the binding site where it can't make good polar interactions, it pays a desolvation penalty. The uncharged water doesn't pay that penalty because it doesn't have any charges. And so in those locations, the uncharged water is more favorable than the charged water. So if we look in a protein binding site, and as an example of these sorts of sites, we can actually get at the individual orientations. Because we've sampled the orientations so thoroughly, we can see which specific orientations are more probable from the ensemble of orientations at that position. And we see sort of exactly the behavior that we would expect. So in the places that are yellow, which are places where there are good specific polar interactions for water to make, there's usually only one or two dominant orientations for water at that position. Alternatively, in places where the uncharged water is more favorable, in the purple positions, water here is disordered, right? There's no good polar interactions to be made, and water is paying a desolvation penalty to be in this place where it can't even find good polar interactions. So we've done the systematic sampling throughout the entire binding site, but here I'm only showing you a small subset of these. And this is because we're showing the significantly strong interactions, both in the negative and the positive direction. And we developed a sense for what's significant by looking at these values over a large number of structures. We ran these calculations over the Iridium set, which is a high quality set of over 120 structures developed by Greg Warren, where the data has been verified and re-refined. And so now we can have some sense of what are particularly interesting values and particularly interesting sites to pay attention to. So if we compare this map of the binding site environment that we get from SZMAP to a ligand that comes in and binds in this site, we see that ligands are often very good at exploiting the environment of the binding site that we can see through the SZMAP calculation. So in places where there are good polar interactions, that's where ligands put their polar chemistry. And we often see ligands mimicking the specific dominant orientations of waters that we see in the SZMAP calculation on the empty pocket. Likewise, where we found the uncharged water to be more favorable, that's a beneficial place for a ligand to put its non-polar chemistry that doesn't pay the heavy desolvation cost that a polar water molecule pays. And so it's this kind of analysis that we think is useful to apply information from SZMAP to the problem of drug design. And so to give an example of how we would do that, we're going to look at this retrospective case of a HSP90 fragment screening hit here and work done by Aztecs. So they found this molecule in a screen, and through a lot of careful synthesis, they were able to improve the binding affinity. And what we want to do with SZMAP is to go back to this original ligand, this original structure, and see if SZMAP can point us in the same directions that they found to be useful through their subsequent synthesis. And so we start with this structure, and we do the full systematic sampling through the entire pocket to get this map of SZMAP information. But because we're interested in the specific ligand, we're going to look in the region around this ligand to see how this ligand is matching the protein environment. And we see some cases like the carbonyl here that's acting as an acceptor right over here, where the ligand is matching the existing environment of the binding site. But there's also regions such as this specific interaction here, as well as this hydrophobic region here that the ligand really isn't taking advantage of. And so those two areas map very nicely to two changes that were found to improve the binding affinity by over 4 orders of magnitude. So taking the methoxy group and changing it into an isopropyl to access the hydrophobic area, and adding a hydroxyl to the bottom here. So SZMAP was able to at least guide us in the same direction, or into these same areas where beneficial modifications were made. But remember, this was done by doing this full systematic search of the entire binding site. But because we can sample using any approach that we want, there's more optimal ways to get at this information. And in fact, very early on in SZMAP, we included the ability to do a very rapid evaluation of the binding site environment by doing the calculation only at the coordinates of the heavy atoms of the ligand. And we included a tool called SZMAP report that will allow you to take the results from one of these calculations and generate this nice 2D depiction of the molecule with the SZMAP values portrayed on top of the depiction. And so out of this idea and this ability to sample at arbitrary coordinates grew the idea for GAMEPLAN, which is an accompanying program that comes with SZMAP, and it's designed specifically for the situation where when you're working from an existing ligand scaffold. And so we'll take this scaffold, run some calculations, and suggest two kinds of hypotheses to you. First, to look at the existing scaffold atoms and analyze how those atoms and that chemistry matches the protein environment at those coordinates. And then secondly, to look in a small region around the ligand to see, are there places where adding a small substituent might be able to access a new significant interaction. And so for the first part, we do exactly the quick analysis that I mentioned before, where we do the calculation at the coordinates of each of the heavy atoms of the ligand. And then we go on to do a bit of analysis to see how does this environment, as measured by SZMAP, compare to the existing chemistry. Are there cases where there's a match, like this carbonyl that we talked about before? And can we identify potential mismatches where the chemistry doesn't seem to quite match the environment of the pocket? But then we want to look out at adding small substituents. And so for every heavy atom, we look at adding an additional atom or two with a variety of hybridization states and with some sampling around rotors. And we do this for 1 and 2 atom additions all around this molecule. And so what we get from this is we generate a cloud of these chemically intelligent coordinates that all correspond to a position where a heavy atom could get to with a 1 or 2 atom addition to this scaffold. And so then we do the SZMAP calculation only at these locations and then show you the hypotheses where we found a significant interaction at that position. And so by doing this GAMEPLAN analysis on this HSP90 ligand, we're able to suggest the nonpolar modification of the isopropyl group as well as the polar modification of the hydroxyl. But the big advantage of this is that instead of doing this systematic grid sampling, it's a focus sampling, and so it's 10 to 20 times faster than the grid-based calculation. So to show another example of that, I'll look at some recent work that's come out of Heptares, where they've been looking at a series of adenosine A2A antagonists. And they've done there have been several publications lately on this series, including comparing a variety of solvent methods to look at how they compare to the binding kinetics. But we're going to ask a much simpler question: can we take their initial crystal structure and the low affinity ligand, and can GAMEPLAN guide us towards the beneficial changes that they found in their synthesis on this series? And so what we see is GAMEPLAN is able to suggest a lot of these modifications that they found to be beneficial. So GAMEPLAN suggests adding a polar group here, adding a hydroxyl group there improves binding affinity by about 1 order of magnitude. GAMEPLAN suggests adding nonpolar groups at the two meta positions on this ring. So both chlorines and methyl groups were added at these positions, and each of those improved binding affinity by about 1 order of magnitude. And we also think we were able to see some evidence for maybe a more subtle change, where on this right side, GAMEPLAN predicts that there is space around here for a second heavy atom. And in fact, going from a methyl group at that position to a trifluoromethyl improves binding affinity by about half an order of magnitude. So this is another example of how we can take a low affinity initial ligand, and GAMEPLAN can very rapidly suggest, come up with some hypotheses that seem to be validated by the synthesis that was done. So now if we look a little bit farther out from the ligand, farther out from these small modifications, often in crystal structures we see crystallographic waters near the ligand, or even if we don't see crystallographic waters, we know water is around the ligand. And so this is often a question, is how do these neighboring waters affect the binding of the ligand? And if you see a crystallographic water, what does that imply for drug design? Do we need to leave this water alone? Should we try and displace it? And SZMAP can address these kinds of questions by classifying these regions into two categories. Situations where water is actually helping the binding of the ligand by stabilizing the bound complex, or situations where the water is actually working against the binding of the ligand or destabilizing the bound complex. And so we address that by going back to doing a calculation on the entire thermodynamic cycle of binding. So we do a SZMAP calculation on the bound state, the complex, and we do SZMAP calculations for the unbound states. So for the empty protein binding site and for water around the ligand alone in solution. And then we compare these energies. And so if we see the water is better off in the complex, in the bound state, than it is in the sum of the two unbound states, in the sum of the empty protein and the ligand alone, then that water in that position is stabilizing the binding of the ligand, and that energy difference is actually part of the binding affinity. So we see a nice example of that in this Scytalone dehydratase case. So this is a protein that's an important antifungal target in agriculture, and it's a very enclosed binding site, but there's these two regions where we calculate water is stabilizing the binding, and in fact you see crystallographic waters at those positions. And this ligand is fairly potent, about 0.2 nanomolar. But from a design perspective, when you see stabilizing waters, I think you have two options, right? The first option is to just leave it alone, because this water is already helping the binding of your ligand, and if you disrupt that, it could penalize binding. But if you can understand the interactions of the water and replace that water with the right chemistry, then you're taking a tightly bound water and freeing it back into solution without giving up any interactions. And so we see nice examples of both of these cases in the Scytalone. So here's kind of the reason why you might not want to disturb it is, so when they changed this position, which was a nitrogen, into a carbon, now the hydrogen bumps this water out of its preferred position, and you actually lose a lot of binding affinity just by disrupting this stabilizing water. Alternatively, placing a nitrile group at that position lands the nitrogen almost directly where that water was, which allows it to interact nicely with the tyrosines that the water was interacting with. And here you get a big boost in binding affinity. So that was the case where the water is stabilizing. But you can also have the situation where the water is not as well off in the complex as in the unbound state. So if you sum the unbound states, the apoprotein and the ligand, and the water is better off in the unbound state, then that water in that position is actually destabilizing or working against the binding of the ligand. So notice that this can happen even when in each of the individual states, the water is in a favorable place for water to be, because it's the difference between the bound and the unbound states that determines stabilization or destabilization. So there's a very nice example of this in some of the early neuraminidase compounds. So neuraminidase is a very hydrophilic pocket, and this sialic acid analog places a hydroxyl group near these two positions. And so at first glance, this would seem like a very favorable place for water to be. But when we do the SZMAP calculation, we see in both of these regions, SZMAP tells us that water is actually destabilizing or working against the binding of this ligand. And so from a design perspective, if you see a destabilizing water, then it's clear that you want to change something. You'd like to stop this water from working against the binding of the ligand in some way. And so you could add a modification to your ligand to try and displace that water. But you could also modify the ligand to improve the interaction with that water as it is. So there's an example of that in neuraminidase where they changed the hydroxyl there to a charged amine. So you're introducing charge. This interacts beneficially with the pocket and with an improvement in binding affinity, but we can also see that it improves the interaction with the waters in those two positions. To the point that in the SZMAP calculation, we now calculate that those waters are stabilizing the binding of the ligand rather than destabilizing the binding of the ligand. So again, now we find ourselves in this situation where we have stabilizing waters. It may be best to leave them alone, but if you can understand the orientations of the water, which you can get from the SZMAP calculation, and you can find the right chemistry to replace the specific interactions that the water is making with the protein, then you can displace those tightly bound waters without giving up interactions. And that's exactly what Relenza is able to do. This guanidino group very nicely mimics the dominant orientations of the waters that we see in the SZMAP calculation, and the binding affinity is improved again. So that's a quick look at the way we think of applying SZMAP to the problem of drug design. But this is still a young application for us, and one that we're very excited about continuing to push the science forward. And so we're exploring a lot of different ways to improve, enhance this information, and use it in a variety of different settings. And so one of the things that we're interested in, of course, is to use different probes beyond water. You know, can we use a charged ammonia? Is there any practical use to trying different probes, as well as trying multiple probes? Can we have two explicit waters at a time in the binding site? The combinatorics here can get quite large, but I think we've got the infrastructure in place now that we can begin to handle even that kind of combinatorial explosion. Another thing that we can do is, from the orientational ensemble at a given point, we can actually calculate the effective dielectric of water at that position. And this is really exciting because we know that proteins modify the dielectric in the binding site. And now we have a way to visualize the strength of this modification. And we can think about cycling this around and using it again in a SZMAP calculation, or even in a straight Poisson-Boltzmann calculation, where instead of assuming that the dielectric of water is 80 in the continuum solvent region, we now have a calculation of how that dielectric varies throughout the binding site. So lastly, there's certainly a lot of potential for using the grids, the information that comes out of a SZMAP calculation with some of our other tools in other approaches. So in ligand-based virtual screening, you could think of using SZMAP grids in rocks, or using kind of derived electrostatic information from these ensembles of water in a tool like EON that looks at electrostatic similarity. Clearly, you know, this information could be useful in docking or as some sort of scoring function. And you could even think of using it in a fragment replacement tool such as Brood to do a kind of post-analysis on the new fragment that you're bringing into a molecule. So these are all ideas that we're beginning to consider, or that some of our customers are beginning to explore. So from a more technical standpoint, we're also continuing to push things forward We're very close to a release of SZMAP 1.2. It's in the last stages of quality control. And this is really a complete rewrite of the underlying calculation, which speeds things up. That alone speeds things up, but where we've also implemented full multiprocessing. And this is really important because the individual SZMAP calculations are completely independent of each other. This algorithm is very amenable to a parallel approach. And so we see in our testing that the performance scales very well out to a large number of processors. In fact, we see even, so here's some example timings of a stabilization calculation. So here's with the current version of SZMAP 1.1.1, so it's taking almost 3 hours to do this stabilization calculation. And even just, again, on one processor, we see about a 25 to 30% speed up on SZMAP 1.2, where now we're down close to 2 hours. And scaling up to 4, 8, 16 CPUs, we can get this down to 10, 15, 20 minutes. But we didn't want to stop just at 16 CPUs on one machine. We know some of our customers have much bigger clusters than that. So we spun up a number of Amazon compute nodes, and we're able to scale this very nicely out into hundreds of CPUs, where if you have a large amount of resources available, you can take a calculation that used to be 3 hours and now have it done in a minute or two. So this is very exciting for us in terms of being able to address problems that before just seemed like they might take a little bit longer. And again, all of these speedups apply to GAMEPLAN or to any of the other more focused sampling approaches that we might try as well. So to conclude, SZMAP and its associated program GAMEPLAN are tools for looking at water thermodynamics, but with a strong focus on applying that information to the problem of drug design to give us information about the binding site environment, about how neighboring waters might be influencing binding of a specific ligand in terms of stabilizing or destabilizing the bound complex, and through a tool like GAMEPLAN, by working with a given scaffold to develop hypotheses of modifications that might be beneficial for improving binding affinity. So with that, I'd like to thank you for your attention and your patience with our problems with the audio, and I guess we might have time for a very short number of questions since we've gone a little long here.
Okay, thank you, Matt. What I'm going to do is, we've had a number of questions that showed up in chat. I'm going to ask two that I think might be of interest to the audience as a whole, and then we will reply to the rest of the questions that we received by email individually. We apologize, because of the delay, we're not going to be able to cover all the questions that we wish we could have. So the first question is, SZMAP is a discrete method, and in MD methods, you can see things like water chains being shown. So is there a correction in SZMAP for the presence of other water in the active binding site? So can you see other water? Can you correct for that?
Right. Okay. That's an excellent question, and one that I think there's a lot of interest in these water correlations and things like that. So we do address having other waters in a binding site through continuum solvent. So for say this explicit water in this position, we are getting the energy of this water interacting with other waters in, say, this neighboring region through the continuum solvent energy, the interaction of that water with the continuum solvent in the neighboring region. So I think it's an open question as to how much water-water correlation really affects, comes into play in these sort of situations. I think we can make the case that by including the energy of the interaction with other waters, we're getting most of the way to water-water correlation. We don't have the specific, maybe orientational influence of an adjacent explicit water, but we get the energetic influence of it through the continuum solvent. It's certainly something that we want to experiment with. That's why we're pushing towards trying or towards having, say, two explicit probes at the same time. But I think so far we've seen little evidence that we're missing much by using continuum solvent to describe the water-water interaction in a binding site.
Thank you, Matt. So the second question, actually we're going to squeeze 3 in because there is a question about future developments. So the second question is, how do you explain how SZMAP deals with translational entropy?
So right now there is no translational entropy in the SZMAP calculation because the ensemble that we're considering is only the orientational ensemble at a given location. I think the translational entropy information is there in the grid when we do the systematic sampling. You could think of looking at neighboring points or trying to do an integration of a well, for example. That would be one way you could think of getting at the translational entropy. So far, we have not attempted to include that information in the SZMAP utility, but I think it's there in the grid.
Okay, and the last question we have time for today is there any plan at OpenEye to be able to export grids to other applications to visualize? Okay,
yeah, that's an excellent question. So that is something that we make possible for users. So by default, output from SZMAP is an OpenEye binary file with the grids attached to the molecules. But we include some command line tools with the SZMAP distribution. One of them is called SZMAP Grid that allows you to take a SZMAP output and extract from it individual grids of interest and write them into a variety of grid formats, CCP4 grids, ASCII grids, Delphi grids, and so using those tools, you can extract a grid from a SZMAP output and get it into a format that you could then open it, say in PyMol or another viewer like that. So I think that's even discussed in the documentation. There's a little workflow for extracting a grid into a format that's amenable to other programs.
All right, thank you very much. Thank you for your patience with us. I wish we hadn't had the audio difficulty because we could have covered all of the questions today, but unfortunately because of that, we are going to have to cut the question time off. Thank you again for attending. Let me note that there will be a link sent out to you sometime in the near future. So if you missed part of the presentation, you can go back and listen to that. Appreciate your time and attention and the questions, and thank you very much. Bye.
Thank you. Bye.